Hi, welcome to Biotechnology 3. This is Jamie Abdullah, and I am going to do just a quick walkthrough of this course with you. So, Biotechnology 3 is a unique course. It is the second year course of the biotechnology sequence at Carlmont High School. This class is very heavy in plant biotechnology. And so you'll see as I go through that there's not a whole lot of modules. And that's because each lab will take anywhere between 7 days to up to a month. And so students are going to be responsible for collecting data, they're going to be responsible for keeping their plants alive, and then when they're all done, they're going to do analysis. Students also demonstrate their understanding through conclusion write-ups. And most exams we give are going to be like practical exams. So you're not going to see a lot of quizzes except for a lab safety quiz in this course because it is a hands-on course. So we're going to start here with our welcome page, telling students all about what we're going to be studying this semester. Let's see, telling them about the Canvas calendar and how it's going to be up to date. If we go next to the syllabus, so the navigation bar, I took your advice and I sort of organized it in the units that you'll be, or the modules you'll be using the most are up at the top. So my syllabus here is linked with a live link, and that way if I have any changes, it will be immediately reflected and I don't have to update the PDF. And then this is just sort of, some summary information and the course summary as well. And then we're going to hop into the modules here. And so the students are going to begin with an introduction to plants. And so we have the plant overview here, which just kind of tells them about plants generally. The oldest fossil dates back 3.8 billion years. And in this unit, they're going to be responsible for submitting a germination chamber data, flower dissection, and seed dissection. If we go back to modules, we'll see that I give them a little bit of information about seed germination here, and then the lab is actually here. Same thing with plant structure and function, and then they're going to be doing a flower dissection. I provided a lecture that eventually I'm going to screencast a video in case they didn't take away everything they needed to from my lecture. And then every student is responsible for one flower, and then they're going to need to put their data on this slide deck. And so in the end, we're going to have about 100 different flowers that the students can look at the data and, ask their neighbors or their classmates if they have any questions about that. Okay. And then we go back to modules. Same thing with the seed dissection. They're going to be sharing their data. And then we're going to get into genetics after, I would say, about 3 weeks spent on this unit. In genetics, we're going to review mono and dihybrid crosses, which they covered in biology and AP Bio. We're also going to look at Wisconsin fast plants. These plants take about 45 days to grow, and then you can harvest the seeds and grow a second generation. So this is going to be one of those long-term labs. We're going to look at pollination techniques because the students are going to be responsible for pollinating their Wisconsin fast plants. And then in countries such as China, they're experiencing a significant decline in bees. And so we're going to look at how those farmers are dealing with that. And we can have a conversation about that. So Oompa Loompa Genetics is just, genetics review. And then I put in some review videos here in case they forgot mono, dihybrid, and non-Mendelian traits. Wisconsin fast plants are going to have to collect data, so they have to plant their seeds. There's a video in here in the slideshow. Oops. In here in the slideshow. And then they're going to do some research on pollination. They're going to be doing a discussion and the birds, the bees, and the plants and the trees. Then we're going to move on to plant propagation. Again, this unit is sort of a work in progress. This first lab is done where they're going to be propagating plants from either stem or leaf cuttings. And then the subsequent labs, I still need to work out the kinks on them, the hormone facilitated propagation and the tissue culture propagation, because these labs are going to have to be done at home and they won't have sterile technique, and we can't necessarily use all the materials we would use in a lab. So these are going to be longer term projects for me where I'm going to have to try doing it at home and waiting for the tissue culture propagation. This is a 6-month long lab. To have the plant large enough to transplant. Hormone facilitated takes about a month. And then the simple propagation, they're going to be collecting data over the course of a month as well. And then monoculture research project. So monoculture is basically planting clones, such as the case with bananas. And so we're going to look at the trouble with bananas. So in the 1950s, one of the most available banana species went extinct because of a fungus, and we're kind of seeing that again with the Cavendish banana. And so I'm going to have the students read some articles about monoculture tied to the Irish potato famine as well, and come up with a list of pros and cons. And so, you know, having a banana that tastes great reliably is a pro, but, you know, one fungus could wipe out your entire crop is a con. And then they're going to apply that, that knowledge that they've gotten to the Elkhorn Slough, which is right outside of Watsonville. And so they're going to be coming up with a PowerPoint presentation. They're in charge of 50 acres of land, and they need to decide what they're going to do with it. Are they going to plant? Are they going to use monoculture? Are they going to save some of it to be protected for animals? And then they're going to narrate a PowerPoint presentation, and then they'll share that. It has not been fully fleshed out yet because normally they do this in class, but I'm going to come up with some sort of system. And that's about it. So I appreciate any feedback you have. Quizzes are here. Discussions are here. Grades are here. So the tools they'll be using most in the navigation bar are going to be right up front and center. All right. Thanks very much for your time. Goodbye.